Welcome everyone. It's my great pleasure to introduce Damien Marie, who is a research staff scientist at the CIBM MRI niche section, and he will be hosting and chairing the session today. Thank you.
Good morning, everyone. So today it's my great pleasure to introduce Lucas Peck on behalf of Patrick Louis-Lemier. So Lucas did a bachelor in psychology and a master in cognitive neuropsychology at Vrije Universiteit Amsterdam. Through this master, he also did a stay at McGill University in Montreal. Then he moved to Geneva, where he did his PhD with Patrick, that he completed a few months back. And on the use of neurofeedback in a very original manner, because with Patrick, they use neurofeedback as a novel method of investigation of cognitive function, in particular face processing. And he's also very much in favor of open science. As part of his PhD, he was also involved in the optimization of an open NFT, of the software Open NFT, which is a software to run neurofeedback experiment. So Lucas, the floor is yours.
Thank you. Thank you, Dan, for the introduction. Hi, everyone. So yeah, my name is Lucas, and today I'll be presenting two projects that we conducted in the context of my PhD, where we used fMRI-Neurofeedback to investigate human face perception and also to modulate it where we could. So before I get to the studies, I want to talk a little bit about the method, fMRI-Neurofeedback, and face perception. What do I mean by it? So starting with the method, fMRI-Neurofeedback at its core aims to improve cognitive functions or clinical outcomes. And we do that by training participants to modulate their own brain activity by giving them access to that brain activity in real time or near real time. And so what happens, we have participants in the scanner, we acquire their functional brain data, but rather than analyzing those data days to weeks after the acquisition, those images are sent incrementally to another machine that has neurofeedback software running. And this software takes care of all the online processing, online motion correction, real-time signal processing, and also incremental statistical parametric mapping, as you can see here. Importantly, what it allows us to do is to extract the activity of one or more regions of interest, and we couple that activity to a feedback display. And in case of the first study, this display was a very simplified thermometer. So what happens is that it starts to move according to the activity in the region of interest, and we provide that display back to the participant in the scanner. And so at this point in time, he or she has access to their own brain activity. And then we tell them, okay, try to find ways, mental strategies, imaginations, visualizations that you can reliably link to the movement on the thermometer. And so they can use the feedback cues to sort of guide their strategy selection. And because the regions that we targeted were face-sensitive regions, most of those strategies revolved around face imagination. So if the participant is then successful in doing this, then we as researchers are interested to see if this leads to enhanced cognitive performance on some metric, or if it improves clinical symptoms. And the studies that I discussed today are more in the realm of enhancing cognitive performance, face perception in our case. So we use neurofeedback in the first study to investigate face perception. We tried to modulate the activity in two separate regions and see how that affected behavioral performance. And in the second study, we used it to enhance face perceptual abilities, for which we had a behavioral and a functional task to measure this. Okay, so face perception or face processing, I use those terms interchangeably. They're not entirely the same thing, but for today's discussion, it's fine to equate them. So face processing describes the process of how the brain perceives, analyzes, interprets, and responds to faces. And so from this lengthy definition, we can already tell that this is not a single brain function. In fact, it's a culmination of many codependent processing stages, which we can readily learn when we think about all the different kinds of information that we can extract from faces. We can say something about someone's identity, who's this person, who does the face belong to, the gender, the age, but also the emotional state or the mood. So I want to start by giving an example of two such processing stages that are central to the first study and to some extent the second study, face detection and recognition. So from this noisy background image, I slowly reveal to you a stimulus, and I'll pause it halfway. Yeah. I think we can all agree that a face starts to appear here. We see two eyes, a nose, and a mouth, and there's an oval shape. But beyond this, we don't know much else about this face. So we can make a face detection, but nothing much else. We don't know who this face belongs to. So now I continue the presentation and I provide your brain with more visual information, and all of a sudden you can say much more about this face and you can recognize the individual. Maybe you recognize the person to be me. So that's detection and recognition, and face detection is interesting. It's arguably the first processing stage when it comes to face processing, and it requires us to extract the common features of a face. So we need to understand what makes a face a face, and researchers often refer to this T-shaped schematic of a face in the eye, nose, and mouth configuration. And that's a very simple pattern. In fact, any stimulus in our environment that adheres to this pattern has a higher probability of being perceived as a face, which is why we can see faces in constellations of clouds. Maybe you can see it here, or in the trunks of trees, or in houses. So anything with that kind of pattern. So it doesn't require a lot of visual information. It's a simple pattern. Face recognition, on the other hand, is sort of the opposite. We need to engage in fine-grained analysis. We need to understand what makes the face in front of us unique and what sets it apart of all the other faces that we know. And so it requires more visual information. So that's just two processing stages. I want to put them in the larger framework of face processing from an information processing perspective. So imagine this is face processing proper from start to finish, takes maybe about 1 second in your brain. And imagine the following scenario where I present to you the face of Tom, and Tom is a good friend of yours, and you had a coffee with Tom last week. So what might happen? A detection stage is the first one to go. Then when you detect the face, you acquire more detail. You understand that you're looking at your good friend Tom. You have access to a personal memory, and then you come to remember that indeed you had a coffee with Tom last week. And then concurrently, you have all these other processes ongoing. You might want to evaluate Tom's emotional state, maybe where he's looking, how attractive he is, and maybe if you don't know Tom, you want to gauge his attractiveness based on his facial features. So the last 20 to 30 years, a lot of research has gone into face processing. So we have quite a good understanding of the functional neuroanatomy of face processing. And I just want to briefly go over the different regions and networks involved without going into any detail. So here's a brain that's presented with a face. Then what we see pretty much in all individuals, we have these three highly face-sensitive regions that activate: the occipital face area, the fusiform face area, and a posterior cluster in the STS. And one way to think about what these regions are doing is that they provide the brain with a crisp, detailed image of the face in front of us. And then you have all these other regions that get involved secondarily, and they extract the information that they themselves are working on. So collectively, these three regions are referred to as the core face network, and they take care of the visual analysis of faces, which means they are involved in face detection, face feature analysis, eyes, nose, mouth, the visual component of recognition. So they provide the detail that we need. The visual component of emotional expressions. An angry face looks different than a happy face, but also eye gaze, lip movement, and dynamics. So once a crisp image of that face is established, these other regions get involved that are less face specific. I think we can all agree that dorsolateral prefrontal cortex is involved in many cognitive functions or executive functions, but also in face processing. And together, this network is referred to as the extended face network. And these regions take care of the abstract or more abstract analysis of faces, which means they're involved in emotional processing, person memory, the identification part of recognition, trustworthiness, face motion, visual attention, attractiveness, etc. And of course, it's highly interconnected, and it's the network functioning that allows face processing to transpire in your brain. In the first study, we focus on OFA and FFA and their relation to face detection and recognition. And in this second study, we zero in on the FFA and its relation to detection specifically. And then we try to train participants to modulate FFA activity, and we see if that improves their face detection abilities. And so for face processing generally, there's a strong right hemispheric dominance. This is important for today because, as you will see, we targeted only the right hemispheric clusters in the regions of interest that we used during the neurofeedback training. So with that background, let's turn to the first study where we used neurofeedback to investigate the roles of FFA and OFA in face detection and recognition. So why would we do that? Well, if you read the literature, there's a disagreement about how exactly visual face information propagates through the face network, through the core face network, and in particular between the FFA and the OFA. And as a result, different functions are ascribed to these different regions, and that then has implications for face detection and identity recognition. So that's a lot of words. So I'll try to display what this all means. So we have a hierarchical model. According to the hierarchical model, face processing is a local to global phenomenon. We start with the details, and from that, we form a complete percept of the face, which means that face information arrives in V1, first enters OFA. OFA is then the first to respond to face information, is involved in feature analysis, so the eyes, nose, and mouth, etc. So the high spatial frequency information, and therefore can be linked to face detection. As that information propagates through the brain at the level of FFA, we have complete percept of the face, and we have enough information to allow us to recognize an individual. So FFA is involved in recognition. Then there's another model, the non-hierarchical model, which flips it around. They propose it's a global to local processing phenomenon, where we start with the crude representation of the face, and we fill in the details after detecting such a face, which means that face information bypasses OFA and enters FFA. FFA is sensitive to low spatial frequency information, holds a crude face representation, uses maybe this T-shaped schematic to detect a face. And so FFA is involved in detection. And then once a face is detected, other regions are recruited. One of those is OFA. OFA is involved in the detailed analysis of faces, provides that information back to FFA, and at the level of FFA, or potentially more frontally, there's an integration process of these two, which then allows us to recognize an individual. And so according to this model, OFA contributes to recognition, but so does FFA. And then what we did in the first study, we used neurofeedback to train participants to increase OFA or FFA activity. And then we wanted to see if that had differential effects on face recognition and face detection performance. So more formally, we used neurofeedback to preferentially activate FFA or OFA prior to, and then hopefully during, face detection and recognition. And for this, we had a task during which participants had to detect faces and recognize faces. And here is one trial. It took about 10 seconds. So over the course of these 10 seconds, we slowly revealed to the participant the image of a face or of an animal. And they had to decide as soon as they knew which one it was, if it was a face or an animal, the detection response. And then subsequently, they were asked to indicate whether they recognized that image from a previous task or if it was an image they had never seen before. And then crucially, we use neurofeedback to modulate, to increase the activity in one of these two regions. So that's the pre-trial activity. And then depending on which model you consult, you can make different predictions as how that would affect face detection and recognition performance. And so to illustrate, let's imagine that the current participant is in an FFA training session, and on this trial managed very nicely to increase the FFA activity. Now, if we consult the hierarchical model puts FFA second in line, mainly involved in recognition of faces or contributing to recognition of faces. So we would anticipate to see enhanced recognition performance. The non-hierarchical model, on the other hand, puts FFA first as an entry node to detect faces. So we expect to see enhanced detection performance, but also at a later stage, it's involved in an integration process. So it also contributes to some extent to recognition. So we might also expect to see enhanced recognition performance. So we can make similar but different predictions in the OFA scenario. But I'll just skip over that. It's pretty much the same thing. And I hope that now you understand what we tried to do. We delineated our regions of interest at the individual level using functional localizers, and we targeted only right hemispheric clusters. How did we do all of this? We had two groups. We had an experimental group that trained to increase that pre-trial FFA and OFA activity on separate days. And we had a control group that received sham feedback. So these participants thought they were doing the same thing as the experimental group, but unbeknownst to them, the neurofeedback they received, so the movement of this thermometer was unrelated to their own brain activity. So finding any effects exclusively in the experimental group brings down more confidence, harbors more confidence in our neurofeedback training protocol. Receiving real feedback should have an effect, whereas random feedback should have less of an effect. All these participants performed in 2 neurofeedback training sessions on separate days. So on one day, they might train one region, and on the other day, the other region. And of course, the order of this was pseudo-randomized and counterbalanced across participants within each group. So the neurofeedback training, I'm showing you one trial. One trial took about 60 seconds. It has a neurofeedback part and this behavioral part. We did 7 such trials per run. Then there was a short break, and we did 7 such runs per training session, amounting to a total training time of 60 minutes. Now, that's the participant view, what they saw in the scanner, and just walk you through the different steps here so you have a better idea of what we did. During baseline, participants were counting down just to keep them occupied. Of course, we were tracking their activity in FFA and OFA. During regulation, they were presented with this feedback, the thermometer that would now move according to the activity in one of the regions. And they were trying different ways, thinking about faces, visualizing faces, imagining faces, whatever. Hopefully they are successful. And then right after, they will be presented with this task that you're now familiar with, where we show a face or an animal and have to make a detection and a subsequent recognition response. And each trial would then end with another feedback, intermittent feedback score, which summarized their performance during self-regulation. So 2 feedback moments. So what do we find? We have 3 effects of interest. We want to know about the neurofeedback performance, where participants able to increase their target ROI activity, FFA in the FFA session, OFA in the OFA session. Did that modulate how those regions behave during the task? And in turn, we want to know if that had an effect on behavioral performance, detection and recognition, and those are expressed in reaction times. So lower scores mean better performance. So what about the performance? Were participants able indeed to upregulate that activity and the target ROIs? And of course, was the experimental group better at this than the control? So first, let's look at those regions in isolation. We have the OFA on top and the FFA at the bottom, and we're looking at session averages. So here you see the session activity of the OFA during the OFA training, and we compare the experimental with the control group. And we see that activity during baseline, regulation, and task. And we can see clearly that the experimental group outperforms the control group. So receiving real feedback rather than random feedback helped the experimental participants to find strategies that were more effective in activating that activity. And we find a similar effect in the FFA during the FFA training. Again, experimental group outperforms control group. And forgetting about the control group, we can also compare between the training sessions within each group. So here's the experimental group, again, OFA activity. And we see that the OFA is more active during the OFA training than during the FFA training, which again is a sign of modulation and gives more credibility to our neurofeedback training. We did not find that effect for the FFA, unfortunately, for the experimental group. We have some ideas why that is, but we can discuss that later. So those are just two regions, but it's interesting to know what else happened in the brain during that window of self-regulation. And these are the results of the experimental group. We did not find these effects for the control group. You see that many more regions are co-activating beyond the FFA and OFA. And the take-home message here is that pretty much the four-phase network, as well as the extended phase network, were co-activating during self-regulation. And that's interesting because at this point in time, participants weren't seeing any faces, they were imagining faces. So they recruited similar networks that we use when we perceive a face. Now, that's not really revolutionary, but it's still an interesting finding. And it has some theoretical implications for what we try to establish within the FFA and OFA that I will park for today. So that's the session average, but in neurofeedback, we're always very keen to see improvement over time because this, again, argues in favor of a neurofeedback training protocol. So let's look at that. This analysis assesses this. It's a linear mixed model, and I want to point your attention to this triple interaction effect, which is plotted here. We compare the experimental with the control group. On the x-axis, we have the separate runs within each session, so that's the vector of time. And on the y-axis, we juxtapose FFA to the OFA, and we compare the different sessions, and we see that in the experimental group, there's a strong increase in OFA dominance during the OFA training, and a slightly attenuated, but still increasing dominance of FFA in the FFA training. So that's a learning effect, and we do not observe that in the control group. So now that we know that participants were by and large able to self-regulate, we want to know if that self-regulation activity affected how those ROI, how those regions behave during the task, during detection and recognition. And for this, we look at it slightly different. We ignore the control group and we ask, okay, was the activation here, did that affect somehow how those regions behave during the subsequent task? So we have the FFA here and the OFA here, and we found that indeed, both regions predicted their own detection and recognition responses. Now we can argue about the nature of this relationship, but that we found it is important because in turn, these detection and recognition responses have very distinct effects on behavioral performance that are very much in line with one of those two models that I just discussed. So we found that the FFA positively contributes to detection. So that's a negative effect, but these are reaction times. So it means enhanced FFA detection responses were related to lower detection time. So improved performance in FFA contributed to detection as well as recognition, and OFA only to recognition. And so that's in line with the non-hierarchical model that suggests that FFA is indeed first involved in detection, at a later stage in recognition, and OFA only provides the visual information that we need to make to identify a person. Okay, so that was the first study, and one of the findings was that FFA involved face detection, and so then we went on to test this more directly using again fMRI-Neurofeedback to modulate face evoked activity directly in the FFA, and then we wanted to see if that would lead to enhanced face perceptual abilities, for which we had two separate tasks, and this has certain clinical implications, as I will tell you soon. Now, it's important to realize that in the first study we used neurofeedback to modulate that pre-trial activity. And in this study, we tried to train participants to increase activity within the FFA directly related to face perception. So there's no imagination in that sense involved. So we read the title, we use neurofeedback to enhance subliminal perception in the healthy brain. So what is subliminal perception and why did we use it? Subliminal perception refers to unconsciously perceived stimuli by an attending brain. Basically, I show you a stimulus, it can be anything, but let's say it's a face in a certain way. This face information is partially processed in your brain, but it does not give rise to a conscious experience. So experimentally, we can induce this with a sandwich mask paradigm where we show a couple of masks, a phase, in our case, a delay of various durations, and then more masks. And as you will see, the duration of that delay dictates whether you process consciously the phase, whether it's subliminally processed or whether it's not processed at all. So this is a conscious trial, and this is what it might have looked like for the participants. A couple of masks, then there is a phase, long duration, and more mask. Now, the idea is that the phase information arrives in visual cortex, propagates through the brain, reaches phase-sensitive regions, and then ultimately frontal regions. Those frontal regions project backwards, where they augment those cortical representations, and this whole feedback system mechanism gives rise to a conscious experience. And the mask arrives way later in time, and so it does not have a chance to interfere with that process. But what happens now if we reduce that duration? It becomes harder to spot the phase, and if we go full subliminal mode, it's nearly impossible, while the phase information is still embedded in the stream of stimuli. So here the idea then is that that phase information arrives in visual cortex, starts to propagate through your brain, all the same, reaches phase-sensitive regions, but it doesn't reach those frontal regions due to the arrival of the mask. And so there are no backward projections, there's no enhancement of cortical representations with these feedback mechanisms, and so there's no conscious experience of seeing the phase. It does mean, though, that there is still phase-specific information in extrastriate cortex, and that's exactly the signal that we targeted with our neurofeedback training protocol. And then, as I said before, we wanted to see if that would result in enhanced performance perceptual abilities. Okay, but why subliminal detection, subliminal phase detection? This aligns with a broader ambition to use fMRI-Neurofeedback to enhance non-conscious neural representations, which in turn is motivated by this interesting but very debilitating neural syndrome, which is called hemispatial neglect. And I'm sure most people are familiar with this syndrome to some extent. It's the result of stroke in frontal parietal regions. And patients present with an inability to perceive stimuli in half of their visual field, despite the fact that those stimuli are processed in the unaffected visual cortices. And so we know there's a weak signal, but that does not give rise to a conscious experience. And that should ring a bell, because that's exactly what we tried to replicate in the healthy brain with our subliminal phase stimulation. We wanted to elicit weak responses. And then if we can show that neurofeedback can be used to amplify those responses to these unseen stimuli and show that this then leads to improved visual perceptual abilities, then we have a proof of concept for future intervention studies targeting patient populations. So how do we do it? In this study, we had an experimental group that trained to increase subliminally evoked activity directly in the FFA. And we reasoned that this would lead to enhanced processing of those stimuli. And we had a control group that in this case received real neurofeedback from a different control region, the interparietal sulcus. And so they were doing the same thing as the experimental group. And we had different expectations as how that would affect the cortical representations related to the whole paradigm. And I will not discuss the neurofeedback training in detail. I'll just explain now what happened. Participants were in the scanner while they tried to increase FFA activity. And while they were doing that, they were exposed to subliminal phases. So each trial would be about 20-25 seconds. And after each trial, they would receive an intermittent feedback score, a monetary reward score, telling them how much the FFA was activated during the previous trial relative to a carefully selected baseline measurement. So all these participants performed 3 training sessions on 7 days. And of course, we want to know if enhancing that cortical subliminally evoked activity, if that would lead in enhanced phase perceptual ability. So we have a behavioral task and a functional task to assess this. With the functional task, we want to find evidence whether subliminal phases are in fact processed in the FFA, and if they are processed to a greater extent after neurofeedback training. And I will talk a little bit about these 2 tasks now, and I will skip on the neurofeedback training part. So the behavioral task that we came up with is a subliminal phase detection task, and it measures the subliminal detection threshold which is derived from an equation, but in words it means something like the moment at which a briefly presented face, in our case face, starts to enter your visual awareness. How do we measure this in the lab? So imagine I present to you a couple of masks, then a briefly presented face, but before the face I also present to you an auditory tone in headphones that you're wearing. We finished the trial with more masks, and in this particular case there was no processing going on, not subliminal, not conscious, because the mask arrived too closely to the face. Now I ask you the question, did you see a face before or after the tone? And because there was no processing, your performance will be about chance level. You're just guessing. But now if I allow more time for that face information to propagate through your brain, then your performance will start to improve and you perform above chance level. Yet at this point in time, you have a high probability of reporting back to me that in fact you're still not seeing any faces. And so that's of course a sign of subliminal processing. When I allow more, like a lot of time for that face information to propagate, you will see most of the faces and your performance is high. And we can do that for different increments. We fit a function to that data, we find the inflection point on that curve, and then we have arrived at the subliminal detection threshold. And lower thresholds, of course, mean better detection abilities, and then we want to see an effect on those detection thresholds before training versus after training. That's for behavior, but how do we assess if subliminal faces are processed in the brain using fMRI, knowing that our bold response is so sluggish and subliminal presentation is so fast? For this, we exploited repetition suppression, which is the reduced neural activity when stimuli are repeated. And we see that in regions that process that stimulus. And it's believed to be the result of enhanced neural efficiency after the first exposure. Basically, you're presenting the same information twice. So on the second exposure, it takes less effort for the relevant regions to process that information. And this then manifests as a reduced or a suppressed bold response in fMRI. So bringing that to the current context, imagine we have the FFA. Within the FFA, there are a bunch of face-sensitive neurons, and we present a subliminal face to those neurons. This will activate a subset of those neurons. Then we continue the trial, and we finish with a visible target. So this one you would consciously perceive. This phase is different. The visible target is different from the subliminal phase, so it will activate a different set of neurons. Then we can record the bold response to the visible target, which might look something like this. And on a different trial, again, I present to you subliminal phase. This will activate, say, this set of neurons, but now the visible target is the same. And so the same neurons are activated, but to a lesser extent due to repetition suppression or this enhanced neural efficiency. And then we can record the activity related to the visible target, and that looks something like this. And so you see it's suppressed versus, in comparison with the incongruent condition. And that is repetition suppression. And it tells us that the subliminal prime was processed in the region because it had an effect. It was picked up by that region. And so we reason that neurofeedback training should lead to enhanced subliminal phase processing. So the prime should have a greater impact on how the visible target is processed. And so we would expect to see enhanced repetition suppression effects within the FFA after neurofeedback training. So to the results. So what as it turns out, not everyone learns to use the neurofeedback signal. This is a common finding in neurofeedback studies. And in accordance with other practices, we separated those participants that improved over time. So that's the performance and the 3 sessions. And we separated those from those that did not learn over time. And so we reason, some of my analysis are focused just on the learners, and we reasoned that, okay, these are the participants that were able to increase that subliminally evoked activity the most. And so that's where we should see the biggest, the strongest effects. And then sometimes we can contrast that against non-learners to show the effect of training. Furthermore, everyone received an individual learning performance score. So we have good learners and bad learners and really bad non-learners. And we use those to correlate with our phase detection measures. So the behavioral task and the functional task, arguing again that those who performed the best during neurofeedback training should also show the biggest improvement in phase perceptual abilities. Okay, before we get to the effect of neurofeedback training, we first want to know, were subliminal phases processed at all in the FFA? Was there a signal that we could improve on? I'm showing you the whole brain results of all the regions that were sensitive to this incongruent versus congruent condition, so the repetition suppression. So that's a bunch of regions. They're interesting, but I just want to point our attention to the following finding, that we found a repetition suppression effect directly within the trained FFA. So this is a signal that we wanted to see improved after neurofeedback training. Was that the case? For that, we turn to a region of interest analysis. Here I'm showing you the cluster on the previous slide in the right FFA alongside the left FFA. This is the FFA group, the experimental group, only the learners, and we have the repetition suppression effect on the y-axis. We see that before and after training, and we see that the strongest repetition suppression effect can be found in the right trained FFA after feedback training. And the IPS group, the learners did not show those effects, not in their FFA, nor in their trained IPS. Then coming back to the FFA group, we also, and putting everyone together, so not just the learners, but also the non-learners, we also found that neurofeedback learning, so those individual learning performance scores, were related to subliminal phase processing as measured with this task, in such a way that better learning was related to enhanced subliminal phase processing. And importantly, the IPS group did not show that effect. If anything, it went the other way. So to the behavior, I'm just showing the results of the FFA group. Here are the group results. I fitted those psychometric functions to the pre and the post measurements, and we see here the detection thresholds. And there indeed is a drop in performance at the group level, which might look something like this. Now you can argue this is maybe just practice effects because they've been exposed to subliminal phases for days now. Of course, they're going to get better, and this is true. In fact, the IPS group showed a similar decline, improvement, but only the FFA group, we found that the degree of improvement, so that's the improvement, right, is related to, again, neurofeedback training, in the way that those participants that learned to increase the subliminal signal the most during training also had the biggest improvement pre versus post in phase detection measured with this task. Okay, so I'm almost finished. I'm going to wrap it up. We found that in this last study that FFA neurofeedback training seemed to have enhanced neural processing of subliminal phases. At least we were successful in the neurofeedback training. And we also found evidence that it was related to improved subliminal phase detection behaviorally. And another effect that we found, but that I didn't discuss, is that subliminal, that enhanced, the subliminal phase processing was measured with a repetition suppression task, was related to improved subliminal phase detection behaviorally as well, so closing the loop. And our control group, the IPS group, did not show any of these effects. So to conclude, we used real-time neurofeedback to modulate subliminal brain activation related to visual stimuli. The FFA targeted training was related to enhanced efficiency in processing subliminal phases. That's one way to interpret it. And this was then related to improved phase detection performance post-training. This has translational potential for chronic spatial neglect patients, which present with a similar neural consciousness disconnect. And so now we have a foundational proof of concept for future therapeutic interventions. And I'm happy to say that in the lab, the next generation of neurofeedback studies are on the way. So hopefully at some time in the near or semi-near future, we can move it to a patient population. And then general conclusions of what I talked about today. In study 1 and 2, we provided evidence for non-hierarchical phase processing, showing that fMRI neurofeedback can be used to regulate two distinct cortical regions involved in the same functional process, phase processing. That's a novelty. Usually regions are selected that have nothing to do with each other and sort of try to find differences there, but we try to do it within the same faculty. And we show that we can use it to enhance non-conscious brain responses to unseen stimuli, which has implications for neglect, as I discussed. So with that, I want to thank you for your attention and everyone on this slide.